Okay. I'm a plant physiologist. I started here in the ARS last April. So I've been here for less than a year, and I'm really enjoying it. It's been even with all the craziness of 2020, it's been a great time, and I've enjoyed learning about sugarcane and starting my research. Um, and it's also, I must say too, it's nice to be back in the southern part of the United States. I'm coming from Washington State, and quite frankly, I was getting a little tired of the cold, so it's good to be back in warm country. Uh, so today I'd like to just talk briefly about my research focus and also some of the research that I'm currently doing. As the title states, across spatial scales, an integrative approach to understanding sugarcane physiology. I uh, I work across spatial scales with a focus on whole plant physiology, and therefore looking at how the different spatial scales affect physiology. And I hope to show you that how on the fine spatial scale, genetics and leaf and root physiology, and also on the field scale, the large spatial scale, and how that's those two scales are linked together through whole plant physiology. So I just want to show a schematic just to kind of illustrate this, and I'd first like to show some of the things that we can learn from each of these spatial scales. And so on the root or and leaf scale, we can there's a focus on physiological and biochemical pathways of photosynthesis, and with roots, we have a focus on uptake kinetics and root growth patterns. And there's a lot that we can understand, and we can understand about plants, and in this case, sugarcane, from looking at these leaf and root levels. And then also, then you have on the field or crop level, we can look at CO2 uptake by photosynthesis and water loss from evapotranspiration, and we can relate that to biomass and yield produced relative to the water inputs to the field, and we can start to understand at that field level what's happening. But and what the link is between these two scales is whole plant physiology and plant behavior, such as the coordination of above ground and below ground organs that relates nutrient and water uptake to photosynthesis and growth, or also like canopy structure and how that affects light interception and water loss and carbon loss through respiration. And then also by we can scale these leaf level biochemical models and also root structure and function to the crop or field scale. And we do that through actually we can do that more accurately, more correctly through a better accounting of whole plant physiology. And I would just like to now show, so this is kind of my research focus, what I'm interested in, but I'd like to show a little bit of the preliminary results that I've done so far. The first study that I'm looking at is to measuring water use efficiency at the field scale. Now, water use efficiency is measured as the gross the gross primary productivity per evapotranspiration. And this is a measure of agricultural sustainability with regards to water resources. And using data that Paul White collected since 2000, gross primary productivity and evapotranspiration that he collected since 2017, and he did that using an eddy covariance system, and I used this data to calculate water use efficiency. Now, one thing that's interesting about water use efficiency is it's a method of we typically see it measured more in in water limited agriculture, where it's a method in which to see how much water is being used relative to in relation to how much crop production there is. And it since I've been here, I've been thinking about that. How can we use or apply this water use efficiency to better understand conditions here in South Louisiana that are definitely a water abundant agro-ecosystem. And now I'm starting to get more interested in the development of a concept called the effective use of water, where we consider how to increase sugarcane production relative to the water resources available. And and also to be able to access or to be able to calculate the amount of water that is available to the plants. I actually calculated that from soil moisture data that Paul has been also collecting since 2017. Now, I'd like to show some of the data that I collected, some of the data so far that I have on water use efficiency, and to start out, I'd like to show this first graph here in the upper left. This right here is a typical graph you find when you're looking at field level water use efficiency, where we compare on the y-axis the gross primary productivity to on the x-axis is evapotranspiration, and the line, the regression of this, the relationship is water use efficiency. And as expected, we found a relatively strong relationship between gross primary productivity and evapotranspiration. And it's fairly consistent between years. You see these different lines for each year, and there's quite a bit of consistency between years. But if you look here on the right, this is the relationship between gross primary productivity on the y-axis, and the x-axis is plant available soil water. You see, there really isn't a significant relationship, and what this means is when you compare gross primary productivity not to how much water is actually being used, but to the amount of water that's available to the plant, we find that gross primary productivity is not being limited by soil water. That at least in the years that I've measured here since 2017, which means here, if you look here on the bottom graph, this is the amount of plant available water in the from 0 to 15, 15 to 30, and 30 to 45 centimeters, and even the driest periods in these 3 years, GPP was not limited by water availability, and that is I think was quite interesting, actually, to look at the system and say, because there's two ways to look at it. Say, oh, we don't have an issue with water, but the other question I have is how can is there a more effective way of utilizing this abundant water source? Can we actually be able to more effectively use this water source to produce sugarcane? And one of the things that I'm doing next is to be able to, I'm now looking at is actually be able to measure water use efficiency at different scales, and so I'm going to be able to understand better what's happening here at the field level. And so I'll be measuring water use efficiency now, and at the sub-leaf level, at the leaf level, and then at the whole plant level. And with this, we can compare it to the field level, but then also start to get at this question of how can we increase the effective use of water here in Louisiana. Okay, now I'd like to just kind of shift gears a little bit and talk about another focus of my research and another experiment that I've started since I've been here, and that's looking at root traits and looking at their role in plant physiology. You know, root traits are interesting. I find root traits very interesting in the sense that they're kind of the hidden half of the plant. They're responsible for so much of what is important to sugarcane production. For instance, all nutrients and water are supplied by them, and one thing I'm definitely interested in is the role that root structure and function play in water and nutrient uptake. They're also important in anchoring the plant and that can actually help to reduce lodging. Now, the interesting thing about all this is that even though it's so important to good plant health, little is actually known about the roots. And this is with pretty much all crops, is that we know much more about what happens above ground than we know what's happening below ground. And that presents a problem, is how can we improve and learn more about roots, or how can we be able to improve root growth if we don't understand much about the roots. And so that's one thing I would like to focus on too, is look at the role that root structure and function plays in nutrient water uptake. Now, another aspect of root traits that I'm interested in too is developing phenotyping platforms for being able to phenotype root traits that we can then use, the idea of being able to use and select for root traits in sugarcane breeding. And I'd like to show you some research that I'm doing with Anna Hill's help now. And one of the things that I think is interesting is for the most part, sugarcane's not unique in that very seldom are physiological traits being used in breeding programs, and there's, I think, a few reasons why that is the case. So far, we haven't been using physiological traits for selection. One is we don't understand well enough the traits, and that's especially the case with root traits. And the second is we need to be able to identify quantifiable traits, root traits that can be accurately and rapidly measured and are under genetic control. And the experiment I'd like to show that I've started is looking at, switch slides, is one of the traits that I'm looking at to be able to phenotype is the root primordia and root bands. And also looking not only at the number of root primordia and the density in the root bands, but also the number and density of roots that develop from these primordia. And the reason why is these traits may relate to downstream traits such as rooting vigor, root system development, and also water and nutrient uptake capacity. The problem with just looking at these traits alone in phenotyping is that they are very difficult and time-consuming to measure. And so being able to do this on a phenotyping platform in sugarcane breeding would be an onerous task at best. And so therefore, if we can find good proxies or indicators of these traits, would be very beneficial in phenotyping and would make this possible to use these traits for selection. So in this study, what I did is I used the parent plants used in the breeding program, sugarcane breeding program here in home, and I did that so I can compare a large number of genetically different individual plants to see how consistently these traits differed across varieties. And I measured a section of the root bands and followed that root development after the stalks were marked to see how many of those primordia developed in the roots. And the first thing I wanted to know was, is the density of root primordia on the root band and the proportion of roots that develop from, can that be measured rapidly? So is it phenotypable? Second, are these traits under genetic control? And third, of course, is then can we then relate those to downstream traits that I mentioned before that are important for in sugarcane production. And so for the results, the first result is I've been able to measure this and been able to develop methods to measure this trait relatively rapidly. And the second I looked at is this trait under genetic control? And of course, this is just like the preliminary results right now, but I would like to show that. So let me explain this graph first. This graph is what we call a rank-ordered graph, where the varieties of sugarcane are ordered from the lowest to highest number of roots per centimeter of the root band. And this graph illustrates the relatively large variation that we've been in the root density on these, they're developing from these root bands. And also, I found that there is a significant genotypic effect here. So it appears that this trait is under genetic control, that it isn't simply influenced by the environment. And of course, I'm still in the middle of studying data collection analysis, but I'm also looking at the root primordia to see how they compare as well. And then also in the future, I'll be looking at one, is this trait heritable? So looking at the heritability of these traits, and then also looking at the relationship between these traits and the downstream traits. And looking at, is this actually a trait that we can measure? We'll be able to, because we can measure this very rapidly. Can we actually use this as a proxy for other root traits? But we'll find out. Hopefully, next time I hear talk, I'll have more information about how this is going. But that's all I'd like to show today. And like I say, I'm excited to be here. It's a sugarcane is a great crop, and I'd like to just, in conclusion, just thank some of the technicians that helped with the data collection, also the American Sugarcane League that gave some of the funding for this research. And with that, I'd like to open up for questions.